Okay, today we are going to discuss the male reproductive system, in which we will discuss the cross-sectional views of two ducts, you would say. The one is the ductus epididymis, and the other is ductus deferens. So, as you can see grossly, that this ductus epididymis, which is a comma-shaped structure, it's a longer blind-ended tube present on the posterior aspect of the testicle apparatus, right? And it has got the head, the body, and the tail. The tail portion is continuous with that of the ductus deferens, right? And you know that it's being the internal content of spermatocord, then it has to traverse the inguinal ligament, and finally it enters into the pelvic cavity. Let's find where it goes. Another schematic view to show you the duct of epididymis, and its tail is later on being continued with the ductus deferens or vas deferens. That was a portion which is to show you that the ductus deferens will finally join with the duct of the seminal vesicle. We will discuss it later on. And form ejaculatory duct that is going to open into the urethra. That was the basic concept, to show you the sagittal section, to show you the bladder, the prostate, and the ejaculatory duct. So again, back to the previous diagram. Okay, so you can see that it's a blind-ended tube, and it's being coiled upon itself. When we see cut section, we can observe an oval, round, or so many sectional views. So let's find some histological section to show it. It's a little bit lighter state, but we can see that we observe a single tube being, when it is wrapped and cut into various sections. You have seen a tube being thrown into so many fours, and you cut, you observe various sections. So you observe one portion of it, one cut section, there is another cross section, and there is another, right? And what you see in the inner side, there lies the epithelium. The epithelium of ductus epididymis is pseudostratified column, right? And it's being lined with the stereocilia. As you know, the characteristics of a pseudostratified, that it has got tall columnar cells and the basal cells, right? And you get a collection inside. This is the collection of spermatozoa. And what histological features, in addition to these, are that each section is having on its margins the connective tissue circular layer. These are the connective tissue, smooth muscle layer around these tubular section. They contract and give the contractility to this ductus epididymis, right? You can see here the labeling that these are the spermatozoa, and this is the epithelium lined with the stereocilia, and the smooth muscle fibers are around. So what ductus epididymis has to do? It first stores spermatozoa and then transport it to the ductus deferens. And in addition to this, the epithelial cells, they are supposed to resorb extra fluid, right? And here is the place, right? And here is the place where the spermatozoa, they get mature and they attain the quality of motility. So these are few functional points about the ductus epididymis. Let me find some other somewhat clear picture to make you. This is again a view of ductus epididymis. Yeah, this is a little bit clear one. Now you can see again the cross sections. The same slide picture is now seen as schematically. The lumen is being shown. There is epithelium, which is pseudostratified, columnar ciliated. That's why you are looking into the different level of the nuclei. This is the characteristic of pseudostratified epithelium, you know. Then there lie the stereocilia, very clear in the lumen, the collection of spermatozoa, and around these there lie the smooth muscle circular layer to give contractility. And intervening, you have got connective tissue in between the sectional view of these tubules. Okay, let's see the slide of it. So epithelium, let me make that it has got principal cell which are columnar, and rest of them are basal cells. If you have to define that what epithelial cells line the ductus epididymis epithelium, right? Okay. After that, let me show you the slide rather. Lighter stain, so we need. So again, you observe the connective tissue on the margin that are also entering into it, and you can see various lumens, either rounded, either oval or elongated, right? And intervening blood vessels could be a part. So concentrate that these are the epithelial lining of every cut section of this tubule, which is pseudo-stratified columnar ciliated. And we get the stereocilia inside. Somewhere we can get the spermatozoa collection. And on its outer side being covered, or there is a smooth muscle circular hair on the outer side of these, which is much developed near the tail region of this ductus epididymis, right? Okay, this is on 4x. So you can see the connective tissue intervening and on its outer side. Let's make it focused on 10x. Now the epithelium is much more clear. Can you see the pseudostratified epithelial lining? And even the stereocilia, fur-like appearance inside. That could be some blood vessel nearby intervening in the connective tissue. And this is the smooth muscle layer on its corner. So one of the slides that you can easily identify with the help of epithelium, right? So that is about the ductus epididymis. Now we will discuss the other slide. Let me show you. Okay, we already saw the ductus deferens along with the ductus epididymis, that where the ductus epididymis tail was ending. There is another coiled tube that was ductus deferens or vas deferens. So contrasting features are epithelium is same. Epithelium that you observe in the ductus epididymis, what was it? Pseudostratified. Again, it is there. But the thing is that it has got a very thick wall and a narrow lumen. Can you see that? So we can distinguish this from other, you would say, tubular structures. Like if we can distinguish it from the appendix, what it has? It has got again a lumen, starry lumen, because of the longitudinal muscular folds are there and giving the lumen a star shape appearance. But in appendix, we observe in the lamina propria the larger sized lymphatic nodules. Definitely, they are not here. Okay, so you have to observe that there is a wider, broad, thick muscular wall and a narrow lumen. Now we have to enlarge, but before going to it, let's mark it. This is the epithelium, which is darker here, and just next to it, the lamina propria. So we consider this as a mucosa. Next to it, we have got the muscularis layer. From here up to here, the muscularis layer consists of inner longitudinal, the middle circular, and again outer longitudinal. Again, the variety is smooth muscle. So that is a wider one. And when we get out of it, we have got the outermost adventitial covering, right? And another feature in this ductus epididymis, grossly, that when it is going to terminate, it has got a dilated portion called as ampulla. That is being showing various muscular folds inside and have got various glandular crypts or diverticuli inside, right? The epithelium in that ampulla changes to locolumnar, but here it is the pseudostratified. Let's take it to the 10x, because this is again a differential tissue you have to learn. So again, concentrate the epithelium here, that is pseudostratified columnar with stereocilia again having the columnar cell and the basal cell, and this is due to the folding of the muscular layer outside. This area is just the lamina propria, and there is no submucosa, right? Just next to it, we enter into the inner. This one is the longitudinal layer, and this portion is the circular layer. And if I rotate it, I keep on going. After this thick one, when there is a change here, this is the outer longitudinal one. And finally, we come across with that of the adventitial tissue that can address some blood vessels. The adipose tissue means neurovascular bundle will be the part of the adventitia on the borders. So again, from outwards to inwards, the outer longitudinal, the middle circular, the inner longitudinal, then comes the lamina propria, and this is the epithelium and the lumen, which is showing the pseudo-stratified epithelium in it. So I think this is all for today. Thank you very much. लेकिन उसमें आपने देखा था कि उसका ल्यूमन थोड़ा इससे और थोड़ा सा वाइडर था, इसी तरह वो थिक वॉल थी, लेकिन एपिथेलियम उसकी थी सिंपल कोलमनर. ठीक है. सो देयर इज अगेन फ्यू डिफरेंसेस. इफ यू गेट इट कि कोई और ट्यूबुलर ल्यूमन हमने देखना है तो कैसा होगा. कुछ और भी आएंगे द नेक्स्ट कमिंग द प्रैक्टिकल्स लाइक यूरेटर. Then we will pick that it should be having the urothelium that will distinguish it from the this was difference. That's all.